Right. So today we have the last part of uh, you know we have the last part of our respiration chapter left, and we will solve some interesting questions that were asked from 2019 onwards. Okay, so very important questions. It might look very easy, but they have been asked several times. So let's head on to that. I think I can start my class since you have not mm, you know pointed out any technical issues. All right, so let's go. So in the last class we just studied about the aerobic and anaerobic respirations, and we completed aerobic respiration. We understood what would be the output of the aerobic respiration, correct? So with that, we understand that aerobic respiration is more efficient compared to that of anaerobic respiration. And then we also calculated the output, that is the balance sheet of the respiration, correct? So this is where we were. And today we will try to understand how do we categorize our process of respiration. Okay. See, if I take all the living organisms and in this living world, we know that carbohydrates are a very go-to material. We can call it as respiratory substrate. Okay, when I say substrate, okay, substrate, especially respiratory substrate. Substrates are the ingredients, or I can tell the molecules or compounds that are used for the process of energy production during the process of respiration. Okay, so when I say a respiratory substrate, we have already studied some of the substrate. For example, one best example that we looked at is glucose. So in glucose is a carbohydrate, and it is a go-to material for production of energy. Okay, where now we can also see another respiratory substrates like fats and proteins are also used by living organisms for the process of respiration. Okay, so give me a second. Yeah, students, I'm really sorry for this. Okay, so let's get back to this. Okay, so when I talk about the respiratory substrates, apart from glucose, our body also uses other substrates like fats, lipids, and proteins also. So first, let's understand how the fat utilization takes place in our body, and then we'll go for the proteins, and then we'll try to understand if respiration is a catabolic or an anabolic process. Okay. So if I take an example of a fat, okay, let's say that there is no carbohydrates and our body is utilizing fats. For the fat to be utilized in our body, first the fats are broken down into fatty acids and glycerol. So fatty acids are utilized in a certain way, and glycerol is used in a certain way. I know that it might look quite complicated, or it might look confusing for you. Like, why did fatty acids and glycerol come into first place when we are talking about fats? See, when we talk about fats, the fats are basically a representation of like this. Okay, they have a glycerol. Okay, to this glycerol, this is three carbon atoms are there, correct, right? So glycerol has three carbon atoms. So to the first carbon atom, let's say that there is a chain of let's say hydrocarbon chain like this. Okay, and then this is called as fatty acid chain. A glycerol and a fatty acid chain will be yielded when you look into a fat. Okay. So in that we have monoglycerides, diglycerides, and then we have triglycerides. Okay. And they all come under fat. So whenever we talk about triglycerides or monoglycerides or diglycerides, which are fats, basically, when I say it's triglycerides, it's nothing but fats. So if I say this triglycerides means, that means all the three carbon atoms will have three chains like this. Okay. So one, two, three, that will be triglycerides. Okay. If I break this down, it will give me a glycerol and it will give me a fatty acid. Okay. Do you understand, right? So now, if I see how these individual components are utilized, each of them are used in a certain way. For example, we have glycerol, and for the utilization of a glycerol, during our understanding with respect to glycolysis, you might have seen this. Okay, let me take you back there so that you will get it. So when we're talking about glycolysis, look here. At a certain point, you talked about dihydroxyacetone phosphate. So how did it come in the first place? So that is where the answer comes here. Okay. So this glycerol had to be converted into a dihydroxyacetone phosphate. Once glycerol is converted into dihydroxyacetone phosphate, again it's interconverted into glyceraldehyde 3-phosphate, and the rest of the chain of the glycolysis continues. That is glyceraldehyde 3-phosphate, and then you have, see, we have glyceraldehyde 3-phosphate, 1,3-biphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, and then phosphoenol pyruvate and pyruvate. So this is the rest of the chain of the glycolysis. So that continues from there when we talk about the utilization of our glycerol. Okay, fats, when it's broken down, it gives rise to fatty acid and glycerol. The glycerol that we have is converted into dihydroxyacetone phosphate, which is in turn converted into glyceraldehyde 3-phosphate, eventually to give rise to pyruvic acid. And you know what happens once we get the pyruvic acid? It gives rise to acetyl-CoA. It enters into Krebs cycle, and then it goes into electron transport chain, and that leads to the production of ATP. Correct? Now, the next part is what happens to the fate of the fatty acid. The fatty acids that are produced are directly converted into acetyl-CoA. And again, acetyl-CoA, you know that it goes into Krebs cycle, and then it goes to electron transport chain and for the production of ATP. Done? So we understood how the fat utilization is happening in our body. Whereas when it comes to proteins, proteins are not the direct substrate. So protein have to be broken down into peptide chains. And the peptide chains, when it's further broken down, it gives rise to amino acids. So we know that amino acids are the basic monomers of our proteins, right? So you know this already. Now, amino acids are of various types we understand. Based on the structure and the complexity of the amino acid that is being utilized, eventually the amino acids are converted into pyruvic acid to acetyl-CoA, and acetyl-CoA goes to Krebs cycle and etc. Okay, so now what did we understand here? We understood that our body uses more than one substrate for the production of energy. So when I say more than one substrate, that includes our glucose, and that includes our that is carbohydrates, fats, and proteins. And each of them have their own way. And eventually it all leads to the same point where is it either it is pyruvic acid or it is acetyl-CoA, and then that leads to the production of ATP. Okay. Now the important question is our respiration a catabolic process or an anabolic process? Now, when we talk about glycolysis, so glycolysis, where we started with glucose, that is C6H12O6, and then by the time we ended the process of glycolysis, it resulted in 2 molecules of pyruvate. And if you look at the structure of a pyruvate, the pyruvate had 3 carbon atoms. The glucose had 6 carbon atoms, correct, right? So whenever a complex compound like glucose is broken down into simpler compound like a pyruvate, this is nothing but catabolism, okay? This is nothing but catabolism. So here also, the fats are very complex like triglycerides. When they were broken down into simpler forms, it gives rise to fatty acid chains and glycerol. And then again, the simpler forms would be acetyl-CoA or pyruvate. Okay, so here also it is catabolism. Now, why do we even have the confusion to categorize the entire respiratory pathway as catabolic or anabolic? Now, the answer is, if you look at how does the production of the fatty acid takes place, where did the fatty acid come in the first place? Our body, to synthesize fatty acids, it directly uses acetyl-CoA. You see my point? So for the synthesis of proteins, our body uses amino acids. So whenever for the synthesis of complex materials to be available, like fatty acids or proteins, we have to construct it from the simple compounds. So we combine several amino acids to produce polypeptide chains, and polypeptide chains will give us proteins, which is nothing but anabolism, right? Which is nothing but anabolism. So that means simpler to complex. We are building something. Okay, just give me a second. Anabolism. So this catabolism and anabolism can be clearly seen as the two phases of the respiration. We can't just tell that, okay, respiration is catabolic because we are just converting these complex forms into simpler forms. Or you can't just say it is anabolic because we are just eating food, we are making into simpler forms, and simpler forms are again constructed into proteins or lipids, etc. Respiration includes the combination of both catabolism and anabolism, and that is an evidential thing that we can observe in the process of respiration. So if a question is asked, is respiration a catabolic, an anabolic, or amphibolic pathway, or none of the above? Your option should be respiration is purely an amphibolic pathway. What is amphibolic pathway? Amphibolism represents both combination of catabolism and anabolism. So respiration shows both catabolic and anabolic characteristics. That is why we call respiration as an amphibolic pathway, not as catabolic, not as anabolic. Remember this, it's a very important point. Okay. Several times simple question like this has been asked, what is respiration? Is respiration a catabolic pathway or an anabolic pathway? And many times we fail to answer this. Okay. It is an amphibolic pathway. Okay. Simple. So the next topic is respiratory quotient. So we can calculate the amount, you can tell, let's say, what is the output? For example, respiratory quotient defines, for example, if I take glucose metabolism, so during the breakdown of the glucose, I utilize equal amount of oxygens and liberate equal amount of carbon dioxide by the end of the process. So this determines about how the utilization of your a substrate, respiratory substrate happens. So is respiratory substrate completely utilized and burned for the production of ATP, or is there a partial breakdown of your respiratory substrate? For example, as I told you, in glucose there is complete breakdown. Whereas if you take about fats and proteins, it does not give rise to complete breakdown of your respiratory substrate. So that is why respiratory quotient is an equation that gives us an idea about how the respiratory substrates get utilized. Okay. So to calculate this, we know that in the process of respiration, oxygen is consumed and carbon dioxide is liberated. So that we can divide it on based on the volumes. For example, if I metabolize glucose, so if I have to calculate the respiratory quotient of a glucose molecule, then the equation would be volume of carbon dioxide liberated during the process to the volume of oxygen consumed during that process. So that is nothing but respiratory quotient. Now, this respiratory quotient is a very important number, and it is a very important determinant to understand the characteristic feature of a substrate that is being used in the process of respiration. As I told you, if I take glucose as an example, right, the glucose goes into complete breakdown or the burn down, I can tell. See, the formula of the glucose is C6H12O6, and it combines with 6 molecules of oxygen to give rise to equal molecules of carbon dioxide, 6 molecules of oxygen to 6 molecules of carbon dioxide. So that shows that it's been completely utilized. So that gives us to 1.0. See, the respiratory quotient for the glucose is 1. Remember this. Whereas if you consider the same thing for the fats, okay, if you try to apply it for the fats, a simple fat would have the formula like palmitic acid. Okay, remember the name. The palmitic acid is an example that we have taken for the fats. Okay, so the formula for palmitic acid is C16, H32, and O2. So the number of oxygen molecules that are utilized in this process is 32 oxygen molecules, but we liberate out only 16 molecules of carbon dioxide. Okay. So now what happens to the respiratory quotient? If you observe, it comes to almost 0.7. It comes close to 0.7, people. So here, 16 molecules of carbon dioxide divided by 23 molecules of oxygen will give us 0.7. Now, this shows that the respiratory quotient has decreased because we are using more oxygen and liberating less carbon dioxide during this process. Okay. Whereas if we take proteins, and the example of protein that we are taking is albumin. Okay. The albumin protein consumes 77 molecules of oxygen and gives rise to 63 molecules of carbon dioxide. And eventually, what do we get as a respiratory quotient? It is 0.8. That is why when you eat protein, you get like little bit of energized. Okay. See, when you take carbohydrates, it is very quick energy and your body burns it very efficiently and your body energizes very soon. Whereas when you talk about proteins, proteins give you energy and it also builds you muscles, okay, a little bit higher compared to that of your fats. Fats have very low respiratory quotient. So now what is important thing for our examination is remember the name palmitic acid. What is the respiratory quotient of palmitic acid? 0.7. What is the respiratory quotient for proteins? 0.8. What is the respiratory quotient of glucose? 1. So these numbers 1.0, 0.7 and 0.8 has to be in your memory until your examination. I bet at least one question is coming from this point, okay, regarding this chapter, okay, 100% I can tell you. Next. Thank you very much. And we end this chapter. We have finished the syllabus of this chapter. So let's try to solve some previous year questions, okay, and see what is our understanding, okay. Chalo. So the students from the YouTube, you can answer your questions or try to answer this question in your chat box. And the students from Zoom, I will launch a poll. Okay, you can use the poll to answer your questions. Okay, just let me look at the YouTube and see the status. All right. Okay. So YouTube students, you actively participate because I can see your response and then I can understand if where you went wrong. All right. Sure. So let's start answering. All right. So all the questions that we have taken is from 2019 onwards, as I mentioned. So let's go for the first question, students, without delay. Given below are two statements. One is labeled as assertion and the other is labeled as reason. So let's read the first assertion statement. Assertion says ATP is used at two steps in glycolysis. And the reason is first ATP is utilized in conversion of glucose into glucose 6-phosphate. And the second ATP is utilized in conversion of fructose 6-phosphate into fructose 1,6-bisphosphate. In the light of these above statements, choose the correct answers from the given options below. Your option 1 is A is false, but assertion is false, but reason is true. Both assertion and reason are true, but the reason is correct explanation of assertion. Both assertion and reason are true, but reason is not the correct explanation of assertion. Assertion is true, but reason is false. Okay, students, you have a minute to answer this question, and I would really appreciate if you answer this question within a minute because it should be very easy for you. Okay, so let me share poll. Let me launch this MCQ poll. Okay, hold on. I should end this poll, I think. Yeah, end poll. Okay, I think the polls are live right now. Okay. And I hope the question is clear for you as well. If you want me to read, I'll read it again. But I would like you to look at the screen and read the question as quickly as possible. The assertion says there are two ATP molecules that are utilized during the process of glycolysis. Okay. And then it says the first molecule of glucose is utilized during the conversion of glucose into glucose 6-phosphate. And the second ATP is utilized during the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Okay. So I hope the screen is shared for you. No? If not, hold on. Now it should be visible. Yep. Yeah. Sorry, students, I didn't observe the screen sharing thing. Now it should be visible for you. Okay. Give it a second. Yeah, there it is. Okay. So let's not kill much time. I will just try to help you with this question so that the next question becomes quite smoother for us. Okay. So assertion says two ATP molecules are used in the glycolysis. Yes, this is the investment that we are going to do during the process of glycolysis. I told you guys, when we have glucose, right? Glucose, which was, okay, converted to glucose 6-phosphate, involves the input of ATP. We give out ATP so that it results in ADP plus phosphate. So this ADP plus phosphate, this phosphate goes and attaches to the glucose at the sixth position, at the sixth carbon atom, to give rise to glucose 6-phosphate. Okay. Now what happens to this glucose 6-phosphate? It gets isomerized to fructose 6-phosphate, right? It gets isomerized to fructose 6-phosphate. Now once this is done, again what do? We add one more ATP molecule. And then what happens to this ATP? It gets broken down into ADP plus phosphate. And this phosphate molecule is attaching the first carbon atom of the fructose, okay? So that becomes fructose 1,6-bisphosphate. And this is exactly where, out of the whole glycolysis process, we know that the glycolysis ATP production is 4 ATP. But when we talk about the net ATP of glycolysis, okay, glycolysis, that is equal to 2. Why do we call it as net as 2? Gross is the overall ATP that we get. Net is after investment, what is the return? You are not calculating the investment in gross, but in net we'll minus the investment and we'll only take the profit. So when I tell that net, that means profit. The profit of ATP that we get of glycolysis is 2. The reason is, out of the 4 ATP, that is the overall ATP that we get out of glycolysis, what happens is 2 of them will be utilized during this process. That is glucose into glucose 6-phosphate. Other one is fructose 6-phosphate into fructose 1,6-bisphosphate. Since both other assertion and reason are telling exactly the same, that's why we go for the second option. That is, both assertion and reason are true, but reason is the correct explanation for assertion. So that is option number 2 for this question. Okay. So let's go for the next question, students. Our next question is, okay, oh, 1 second. I will see if the poll is ended. And it's not ended. Okay. Now I think it's ended. How many times decarboxylation occurs during each TCA cycle? Okay. And the options are twice, many, once, and twice. Okay. So just look at the question very carefully. You will understand it. And it's a very easy question. Okay. How many times decarboxylation occurs during TCA cycle? Okay. How many times does it happen? All right. So can we answer this question? How many times does TCA cycle, decarboxylation occur during the TCA cycle? So the answer is 2. Okay, the answer is 2. When does the decarboxylation occur? So what happens during the process of our TCA cycle? We know that acetyl-CoA comes and it gets condenses with oxaloacetic acid to give rise to citrate. Okay, it starts with citrate, correct, right? Citrate is converted into isocitrate. Yeah. Once you have isocitrate, when you have isocitrate converted into alpha ketoglutarate, so during this process, what happens? A decarboxylation process takes place where one carbon dioxide molecule is removed. And other process is when the alpha ketoglutarate is converted into succinate or succinyl-CoA. Let's say it is succinate. Again, here it is one more carbon dioxide is released. Apart from these 2 points, you don't find anywhere where the carbon dioxide is removed out in the TCA cycle. TCA cycle is also called as citric acid cycle. You should remember that. So decarboxylation is a process of removal of carbon dioxide, systematic removal of carbon dioxide through in the structure, molecular structure. So here we remove at 2 points. At what points do we remove? When isocitrate is converted into alpha ketoglutarate And when alpha ketoglutarate is converted into succinic acid, so that is when you remove the carbon dioxide during this process. So if the question is asked how many times the decarboxylation occurs, your answer should be twice. Okay. Don't go for thrice or many or once. Remember the word decarboxylation. Decarboxylation talks about removal of carbon dioxide. Okay. So that is why you have to go for the option 4, that is twice. So can you go for the next question? Come on, students. Okay, give me some encouragement. I need your answers. So let's go for the next question. The next question says, fatty acids are connected with the respiratory pathway through acetyl-CoA, alpha-ketogluteric acid, dihydroxyacetone phosphate, and then you have pyruvic acid. Okay, so this is the question. How is fatty acid is connected to our respiratory pathway? How is it connected? What's the connecting link between these two? Did we just discuss just like 10 minutes back? What is how this particular fatty acid is utilized by our body? Yes. Answer it soon. See, take a minute and try to answer within a minute, and that will give you a better practice to face your examination questions as well. Okay. Can we try to answer this question? Yep. So first we have fat. The fats, we know that it gives rise to fatty acid chain and glycerol, right? Now what happens to this glycerol? Glycerol becomes, it goes through this, glycerol di3 phosphate and 2-pi-phosphoglycerate, triphosphoglycerate, all the thing, and finally it gives rise to pyruvate. I'll write it as pyruvate. And what happens to fatty acid? Fatty acid chain is eventually converted into acetyl-CoA. Directly it is converted into acetyl-CoA. It has a series of steps, but fatty acid is converted into acetyl-CoA, and this acetyl-CoA enters the TCA. So acetyl-CoA becomes the connecting link for the fatty acid and your respiratory chain or respiratory pathway. So our option should be acetyl-CoA. So what happened here? If you did not understand, you remember, go back to where we understood how the protein metabolism and how the metabolism of fats happen. Just now we spoke, and you will understand what I'm talking about. Okay. Now the next question. What amount of energy is released from glucose during the lactic acid fermentation? Our options are less than 7%, approximately 15%, more than 18%, about 10%. So these are your options. I think this should be the easiest question. Okay. So I will wait for your answer. Come on, students. Make it quick. I have a watch, so I can see the time that you take. Okay. Sure. So they're asking about the amount of energy released during the lactic acid fermentation. So lactic acid fermentation is an anaerobic process, anaerobic respiration. So students, listen to this. Whenever you have an anaerobic respiration, remember that it is always low energy. Okay. Low energy process, especially when it comes to glycolysis. In glycolysis, afterwards you have pyruvate, and pyruvate undergoes changes because of the lack of oxygen in mammalian tissues. So the lactic acid, okay, what happens? It has a very low energy thing. So whenever we talk about low energy, that means it produces less than, okay, it produces less than 7% of energy compared to that of aerobic respiration. Okay. So our option for this question is option 1, that is less than 7% of our energy. Okay. Let's go for the next question. What is the net gain of ATP when each molecule of glucose is converted into 2 molecules of pyruvic acid? The options are 8, 4, 6, 2. Okay, so these are our options. So take a moment and try to think about this question. It's a very easy question and many of you might get wrong. That's why I'm asking you to look at the question twice and try to answer this question. What is the net amount of ATP, okay, when each molecule of glucose is converted into 2 molecules of pyruvic acid? Just gave you a difference between what is a net and what is gross, right? Got it, students? Got your answer? Yeah. So to answer this question, first of all, if you look at the entire glycolysis process, in the prior question only we understood that we are investing two ATP molecules at two steps. One is conversion of glucose into glucose 6-phosphate and conversion of fructose 6-phosphate into fructose 1,6-bisphosphate. That means two ATP molecules we are giving from our side. And then where are we getting our ATP? So we get ATP again at 4 points, okay? One through NADP, other one is when we convert 1,3-biphosphoglycerate into triose phosphate, that is where we get 1 ATP. And then we have 2-phosphoglycerate to phosphoglycerate, when it is converted we get 1 ATP. And then again phosphoglycerate, when it is converted into pyruvic acid we get 1 ATP. So here what do we get? We get around 4 ATP in total. 4 minus 2 would be how much? 2, correct, right? Because we invested 2 and we got 4. So the total net was net ATP that we have is how much? It is actually 2. See, remember this. Whenever this net gain is asked, you go for 2. Net gain means after your investment, what is your profit? So I told you we invest 2, we gain 4, we gain 4. 4 minus 2 is how much? 2. This is called as net. If they ask about gross, you don't count the investment. You don't count this 2. So you just go for 4. So since they are asking about net, our answer is option D, that is 2 molecules. Okay. Okay, let's go for the next question. Identify the cytochrome which acts as a mobile carrier for the transfer of electrons between the complex 3 and complex 4. And your options are cytochrome A, cytochrome A3, cytochrome BC1, and cytochrome C. Okay, so these are your options. Let's try to answer this. See, all the questions look easy, isn't it? Because you just studied this chapter very recently. That's why you feel like it's easy. As the time passes, things become volatile. That's why you require regular revision. So let's look at this question. So all we have to do is identify which is a mobile carrier between complex 3 and complex 4. Okay. So that's our question. So if you look at this question, it's from electron transport chain. And we know that electron transport chain had complex 1, 2, 3, 4, and 5. And we know that complex 1 and complex 2, they donated their electrons to something called as ubiquinone. And ubiquinone donated its electrons to complex 3. And complex 3 gives out its electron through this mobile electron carrier called as cytochrome C. The cytochrome C is a mobile electron carrier that is present in the intermediate space. I can just tell that it's not, I'll just write it like this, okay? It is present in the perimitochondrial. It is present in the perimitochondrial space And what is mobile electron carrier? That means it can jump from, or it can move from one complex to the other complex. So it carries the electrons from the complex 3 and it gives out in the complex 4. So that is called as mobile electron carrier, and it is present in the perimitochondrial space or the intermediate space, we can call it as. So our answer is what, students? It is cytochrome C. And rest of the cytochromes are a part of either complex 3 or complex 4. So you just have to look into it. Okay. Let's go for the next question. Very interesting question. The 5-carbon compound formed during the TCA cycle is alpha ketogluteric acid, oxalosuccinic acid, succinic acid, fumaric acid. Oh, the very interesting question, because this question, I will talk about this question first. Try to answer this question, students. Okay. Come on, students from YouTube. Where are you at? Are you able to, are you feeling it difficult or easy? Try to answer this. Don't worry about if it's right or wrong. Right. So usually, some of you might be accustomed to a question: what is a 5-carbon compound that acts as a fixer of carbon dioxide during the photosynthesis? And you write RUBP, right? So usually you might have heard about RUBP. And RUBP, what does it do? It is a 5-carbon compound, ketose sugar. And what does it do? It fixes carbon dioxide. So this is what we have heard in the chapter of photosynthesis. Okay. It is from photosynthesis. But did we hear about a 5-carbon atom formed during a TCA cycle? That is a Krebs cycle. Did we hear about it? Maybe not, but it's time to concentrate. So 5-carbon atom formed during the TCA cycle is alpha ketogluteric acid. It's 5-carbon atom. And remember, succinic acid and fumaric acid are 4-carbon atom containing compounds. And oxalosuccinic acid also, it is 4-carbon containing compound. So that is why the 5-carbon compound that is formed during TCA cycle is alpha ketogluteric acid. So our option is option A. Okay. Let's go for the next question. Which of the following statements is incorrect? So we have to find the incorrect statement. ATP is synthesized through complex 5. Oxidation reduction reactions produce a proton gradient in respiration. During aerobic respiration, the role of oxygen is limited to the terminal stage. The electron transport chain, one molecule of NADH gives rise to 2 molecules of ATP, and one FADH2 gives rise to 3 molecules of ATP. It's a very simple question. All we have to do is find out which statement is wrong. Okay, students, take a look at this question. We have to find the wrong answer. All you have to see is one statement at a time. See if the first statement is correct. It is ATP synthesized through the complex 5. And then you have your second statement, oxidation and reduction reactions lead to the production of a formation of proton gradient in respiration. And you have option 3, where aerobic respiration, oxygen is limited to the terminal stage. And then we have our option 4, where ETC, they say that NADH plus H plus is giving rise to only 2 ATP, whereas FADH is giving rise to 3 ATP. So you see what you can find out here. Okay. So I will help you out. Don't worry. So let's see the first question. The first question is ATP synthesized through complex 5. And this is absolutely right. Why complex 5? Complex 5 has F0 and F1 particles. And whenever we have F0 and F1 particles, F0 acts as a proton carrier pump. So it pumps all the hydrogen atoms from one point, that is in perimitochondrial space, to the matrix. So as it pumps, it goes to the F1, and F1 has what? ATP synthetase. Okay. So it has ATP synthetase. So ATP synthetase, what does it do? It converts ADP into ATP. Okay, thereby producing ATP. So this option is absolutely right. Look at the second question. Oxidation and reduction reaction produce a proton gradient. Absolutely right. So when we talk about complex 1 or 2 or 3, what's happening here? Whenever we talk about the reduction of NADH, or whenever we talk about the reduction of FADH, FADH2, what's happening here, students? So when it is reduced, 4 molecules of hydrogen, and here also 4 molecules of hydrogen was pumped from the matrix to okay, from the matrix to perimitochondrial space, chondrial space. So this variation where at one corner, all these electrons, all these protons are pushed and a gradient is created. The difference in the positive charge is created. So that happened because of the process of oxidation and reduction that was happening in the complexes. Okay, so that's what created this proton gradient. So this option is also absolutely right. And the third option, that is during aerobic respiration, the role of oxygen is limited to the terminal stage. Absolutely right. So we have complex 4. And in complex 4, what happens? Here in complex 4, we have hydrogen. I mean, like, there also complex 4. What it does is it donates the electrons to our oxygen. And oxygen, what it does is it will start accepting hydrogen atom, which is present in matrix. Correct, right? So whatever the hydrogen atom that was present in the matrix, what it do? Oxygen started accepting it, thereby reducing the amount of hydrogen in the matrix. And eventually what happens to this oxygen? It becomes water, which is around 20% of our respiratory output, right? Our body weight. Okay. Finally, what do we get? We get water. So now this is only at the last stage of our electron transport chain. We are using oxygen. Nowhere else we see, not in complex 1, complex 2, complex 3. We only see it in complex 4, which is the final stage where this process ends, right? So that is why this option is also correct. Now, in electron transport chain, 1 molecule of NADH give rise to 2 molecules of ATP. It is wrong. 1 molecule of NADH give rise to 3 ATP. People, it is 3 ATP. And here, FADH2 give rise to 2 ATP molecules. So our option 4 is wrong. All right. So let's go for the next question. You have time? Yeah. The number of substrate level phosphorylations in 1 turn of citric acid is very crucial question and very cunning question it is. The number of substrate level phosphorylations in 1 turn of citric acid cycle is 1, 2, 3, and 4. So these are your options. Okay, it's given according to your options only. So let's see if you can get this right. All right, students. So can we look at what's the right answer? Okay. So the number of substrate level phosphorylation. Where do you see substrate level phosphorylation? I'll give you a clue. Whenever we talk about phosphorylation, that means it has to result in the formation of GTP or ATP, some energy molecules. Okay. So what happens is we have animal cells and we have plant cells. When we talk about animal cells, there is a substrate level phosphorylation where it leads to the production of GTP. Whereas in plant cells, what happens? It leads to the production of ATP. So where does it happen? It happens at only one place. Okay, so that is why our option is option 1. Okay, so if you look at, just observe the citric acid cycle and see where all the ATP or the GTP production takes place throughout the cycle. And you will come across only one place where this is happening. So that is why our answer is 1. Okay, so let's go for the next question. Now, pyruvate dehydrogenase activity during the aerobic respiration requires what? Calcium, iron, cobalt, and magnesium. Okay, very important question. And during teaching also, I've told you that this is very important. Just have an observation. So take a look at this question, people. Pyruvate dehydrogenase activity during aerobic respiration requires what? Which ion? Calcium ion, iron ion, cobalt, and magnesium. What's your answer? See, the questions might look so easy, but can you understand that we have high possibility of forgetting it? That's why people, okay, revise questions also. See, seeing questions also will boost up your memory. Revisit concepts, very must. These 2 units, you take it seriously. Biology is done. Okay. Plant physio and animal physio. Human physio, sorry. Okay, let's see what happens here. See, when we talk about the activity of pyruvate dehydrogenase, where does it come into picture? So we have pyruvic acid or pyruvate. Pyruvic acid plus one CoA enzyme plus NAD plus in the presence of magnesium 2 plus pyruvate dehydrogenase. What does it do? It results in acetyl. Sorry. It results in what? Acetyl TYL, right? TYL, acetyl CoA. So it results in acetyl CoA. So throughout this process, what is used? Magnesium ion is used. That's why we go for magnesium. And our option for this is magnesium. Remember this, very, very important. Whenever you talk about activity of pyruvate dehydrogenase or conversion of pyruvate into acetyl CoA, which ion is required? You go for magnesium. Remember, magnesium, magnesium, magnesium. Okay. Many questions are there. That's why I'm stressing on. Conversion of glucose into glucose 6-phosphate. The first irreversible reaction of glycolysis is catalyzed by which enzyme? Phosphofructokinase, aldolase, hexokinase, enolase. So while discussing this question, like this topic, I told you the one enzyme that you have to remember is this. So the same question is asked here. Correct? Can you observe that pattern? Yeah. So try to answer this. Okay, since we are running a little bit out of time, I will try to rush a little. Okay. Chalo. Can we see which is the right answer? So conversion of glucose to glucose 6-phosphate happens through an enzyme called as hexokinase. Remember these people? Very important. Okay. Because many times, since only few enzymes are mentioned in your textbook, the same questions tend to appear in your NEET examinations. So answer for this is hexokinase. So remember the enzymes, whatever it is mentioned in your textbook, people. That's a clue. Now, respiratory quotient value for tripalmitin is so 0.09, 0.9, 0.7, 0.07. You know what? This was a 2021 question. So it's a 2021 question. That's why I stressed upon this topic while I was teaching in the beginning of this session. Correct? Absolutely right. Your answer is 0.7. Correct. So tripalmitin is nothing but what? It is a fatty acid. Correct. So fatty acid, we know that the respiratory quotient is little bit low. Why? Because they use more oxygen and they give out less carbon dioxide. So that's what is the quotient. And what about 0.8? 0.8 is for proteins. And what about 1? 1 is for glucose or carbohydrates. Correct. So that is why our option is option C. So another question. Which of the following statements regarding mitochondria is incorrect? Our first option is mitochondrial matrix contains single cellular DNA, circular DNA, sorry, DNA molecule, and then ribosomes. And the second option is outer mitochondria is permeable to monomers of carbohydrates, fats, and proteins. Enzymes of electron transport are embedded in the outer membrane. Inner membrane is convoluted with inner folding, okay, or infolding. Such a nice question and such an easy question as well. Take a look at it, students, and try to read the question and answer it. You got it right? All right. Can we look at the right option now? Okay. Which of the following is incorrect about mitochondria? So if you look at the first question, Mitochondria is also called a semi-autonomous organelle. And why? Because it has its own genetic material in the form of single circular DNA. It has ribosomes, it has proteins, etc. So that's why this option is correct. Outer membrane is permeable. Yes, it is permeable to carbohydrates, fats, as well as proteins. Very, very true. Whereas inner membrane is impermeable. Remember that. And next, enzymes of the electron transport chain are embedded in the outer membrane. Now, this is where all the difference makes. It is wrong. It is actually the inner membrane. Okay. Inner membrane. So that is why this option is wrong. Inner membrane is convoluted with infolding. Yes. If you see the structure of mitochondria, you can see that outer membrane is continuous, whereas the inner membrane forms numerous foldings that leads to the formation of cristae, mitochondrial cristae. That is because they want to increase more surface area for our aerobic respiration. Okay. So that is also correct option. So our wrong option is option 3. Now, where is the respiratory electron transport system located in plants? In plants, where is it located? Mitochondrial matrix, outer mitochondrial membrane, inner mitochondrial membrane, and intermitochondrial space. Okay. Where is the respiratory electron transport present in a plant cell? Very interesting question, isn't it? So see how nicely they confuse you. You have an answer for this? Hopefully. Let's wait. All right, students. So let's look at the right option now. Okay. See what if you got the right. Oh, okay. So when we talk about electron transport system, whether it is in plants or it is in animals, okay, animals, electron transport chain happens in inner mitochondrial membrane. The reason they have mentioned plant is because maybe they wanted you to confuse with chemi, osmotic pathway. But whether it is in plants or in animals, the ETC happens in the inner mitochondrial membrane. Okay, sorry, inner mitochondrial membrane. Clear, people? So our right option is option 3. Let's go for the next question. All right, so we have finished all the questions. That's excellent. Okay, so now that leads us to the closure of this session. Please go through as many questions as possible, solve your module questions. Understood, students? So try to solve as many modules as possible, and please ask doubts wherever it occurs and feel free about it. Okay, I'll see you in the next class, and we will discuss about the next chapter, that is transportation. I think, yeah, transportation in plants. Have a nice day, students. Thank you very much for the session. See you.